Hi, and welcome to LabYak, where we interview some of the world's leading scientists and ask them what gets them excited about the science and also about some of their hopes for the future. And I'm absolutely delighted today to be interviewing Dr. Sanjay Singh, who is the Chief Executive Officer of Gennova Biopharmaceuticals, a global company based in Pune in India. Dr. Singh, it's lovely to meet you.
Thank you very much for having me in this part of your interview.
Well, maybe I could start by taking you back. When you were a child, did you ever dream that you'd be a scientist and leader of a global biopharmaceutical company?
Yes, I come from a family which is part of the medical fraternity, and the part of the family was physicist. So yes, there was nothing else in my mind other than being a researcher. But I decided to go into the biology part of that, not the medical fraternity, not the physics, but I wanted to do the biology. Because of how the system works, how the human being works, how the birds works, how the plant grow. Yes, this was a kind of inquisitiveness was there. And so that from very early age, I was fixated. I should work in the area of applied biology.
That's brilliant.
Yes, never thought about that I will be the CEO of a company. But by mere luck or by mere chance, I'm sitting on this position, but still I am a hardcore researcher.
That's great. And did you, I mean, that's really interesting that it started with the inquisition, or the inquisitive mind of how things work around you. How did you choose a university course then? Was it obvious to you that you wanted to go into the biotechnology and biology?
So again, because I did my high school in biology, so there was a path decided that I had to do the biology. So I went for undergrad in biology, and then the biochemistry was very exciting. The university too. I went for a master's in biochemistry, and that slowly opened my path towards my fixativeness, and that drove me to the Central Drug Research Institute, where I did my PhD.
So you went into the Central Drug Institute, and then at some point you end up in America. How did you go from where you were in your studies to ending up in the USA?
So once I completed my PhD, I was offered during a conference a job at New Delhi. So I moved from Lucknow, which is my place of all education, to New Delhi, and joined a center known as International Center for Genetic Engineering and Biotechnology, United Nations Center. And again, this center has been made in Trieste and New Delhi for bringing biotechnology or biotechnology innovation and impact of biotechnology innovation to low and middle income countries. So again, that was a big impact, and I decided to join there, and I worked for a couple of years over there. And I was working in the area of one of the most difficult, protein folding. So there, this was a very complex protein to fold. This has around 12 cysteines. So if you have to fold in one conformation, so you have to avoid 1 to 10 power 12 combination, a billion combination, you have to fold one.
Right.
Took us around three and a half years, but finally we succeeded. But during this process, in one of the Gordon conference at Cambridge, I met the person who was basically the godfather of this area. He said, it is not feasible to fold this protein because it's so many probabilities. And I said, there is no harm in trying. If God folds, you can also fold, only how clever you are close to God.
And how did he respond to that?
So I said, I do not know, but time will tell. And after a year, he was here for the review of my department. And as soon as he saw me, he said, how God is doing. So I said, very humbly, I want to submit that I'm not a God, but I refolded the protein.
For you, fantastic. So he'd come over. What happened next then? Tell me how did he respond to that?
But by that time, due to some personal decisions, I decided to quit. And I was going to one of the top biotechnology company in the West Coast. And when he realized that I had already left the field and I'm going, so he forced me virtually that I should come to NIH for a couple of years.
Uh-huh.
And I said I might change my field because I was working in the malaria biology, developing the vaccines. So I said I may go for a bigger field where things are done a bit differently.
Yeah.
So he said, no. And I think in the next 24 hours, I was having an offer letter from NIH that I should join NIH. Because he was such a humble and great personality, I was not able to refuse him. So instead of ending into a biotechnology company, I ended at NIH.
And the NIH is a place where, I mean, some of the at the forefront, if you like, of some of the protection of certainly of the American public, but also of the global health. And you were there. What was the and timing wise, where were you there before 9/11 or were you there after 9/11? Because the mood of NIH changed, didn't it?
I remember that day of 9/11 because I joined exactly, I know the day, 26 October 2000.
Right.
And I was in a lab and one of my French postdoc came and she said, Dr. Singh, I just heard that a plane has hit the World Tower. And I said, I do not know whether you have got an option to sit into the cockpit of Boeing. You can see 5 miles ahead.
Right.
It is an act of terrorism. And by the time I dialed back home, somebody was on the TV and said, oh, this is the second plane is hitting.
Right. Yeah.
Yeah, that day was very sad, very tough for all of us because that changed our life, how we live, what kind of security environment. But I was in America on that day. Particularly, I remember the incidents minute by minute.
So the NIH mood changed, didn't it, around that time? And I'm guessing that I myself got involved in some of the things around bioterrorism as that period continued, and we did work for the NIH myself. And so I know that some of the priorities that they had changed. Did that change what you had to do?
Yes, there are a lot of things which cannot be discussed, but yes, priorities got changed and changed for betterment because we have to have, because we believe in prevention, that can we prevent something which should not happen, just like smallpox, ricin. So these kind of things were there. The NIH focus a lot in the area of bioterrorism, biodefense, you can say. We heard many organizations come together like BARDA, DARPA, NIH, FDA, CDC, all that kind of things. Yes, but this certainly changed. But my focus area was in vaccinology. So I was focusing in the vaccinology and I continued till 2006 because when I joined NIH, I told my chief that sooner or later I will go back.
Yeah.
And he didn't believe that somebody can have a lab, somebody has a position tenure, and he will leave that. Because NIH position is very unique, that it is indefinite appointment.
Yes.
And you don't have to struggle too much for grants. You don't have to write virtually the grants. So great place, and it gave me a great opportunity it has opened me the whole world.
Yeah.
So it has helped me to think differently, and most important, with such a diverse set of people and diverse set of experience, which has really shaped me for a better scientist and human being both.
Yeah. Well, that's great to hear. And you took that then to Gennova. How did they tempt you back?
So I get very, very soon limited by the environment and I feel I can do even more and more.
Right.
And one of my family friend was on a board of a company, and that company wanted to diverse into biotechnology. And he said that this is opportunities there. And I look into it, talk to the people. This was a very small setup. And then I think I decided this is the point from where I can, because this is a clean slate, can I write the way I want?
Right.
And the way I see these things. This is the part one. The part two is that, yes, a lot of miracles has been achieved by human race in last century. But somehow technologies are so costly that the real benefit of those miracles are not reaching to the most of the needy one. It is limited to the societies of developed world where the cost benefit ratio. So my whole process and thinking was that can we go and innovate and bring the next level of innovation so it can be affordable. And that brought me here because if I worked at one of the best centers in the world, and a lot of experience can be put together with the next setup team with a total different mindset. So can we do afford innovation for affordable healthcare?
And you've made the last 17 years, you've had a very successful journey doing that. And I'll ask you in a moment about COVID-19, because that seems to me to be a really good example of what you're talking about. But prior to that, you managed to launch one of your own drugs. You've a number of biosimilars and a generic. Are they all part of the story then of making healthcare affordable and bringing the technology that you've invented and created and that the world's created to be able to be used by people who can afford those inventions? Is that really what you've been trying to do then for that period?
Yeah, very good question. So from day one, my whole focus on this, that I believe, yes, innovations are very important, but is those innovations are making impact on the society? So when I came as a scientist, which area I should work? This was the question. And this was the basic fundamental questions I want. And you must be knowing Socrates' theory, first principle theory. So what is the fundamental? So we found that one of the disease in India, which is cardiovascular, which is like heart attack, acute myocardial infarction in scientific term. So the drug was available in the West where you have to give it in a bolus and it cleaned the clot within 5 seconds. But this was not available in the country because of cost of affordability. So we said we should develop that because somehow the gene was published pre-1994. And we decided to work on that. So we work on the gene, then we developed the biomanufacturing, which was unique for us. It is perfusion-based bioreactors. So you can basically, you have converted the mainframe computer into iPad. So the kind of innovation. So 5,000 liter you have brought to 50 liter, and you can produce the same product. And today I can say that every month we save more than 5,000 to 6,000 lives by that. And we are doing for tens of years of that. So we bring a smile. In the similar way, we have taken the stroke, where the only drug which was approved last was 1996. So the same molecule we went for clinical trial and we proved the efficacy. And today for that, we are also saving thousands of lives every day. But this process was not easy because sometimes there is a syndrome. If I've not been innovated some specific place, then there is not the innovation.
Yeah.
And which is not true in science, because especially in medicine, it has to perform. It has to be safe. It has to be efficacious. And the last mile is equally important. It has to be affordable.
Yeah.
So we went through some legal battle into it. but I believe in my science. We won that legal battle. And after 5 years or 6 years, once we took the product, now the whole world is shifting to use that particular medicine for the treatment of stroke. So very happy that because I want to see the everyday impact of our innovation. And same way, this COVID-19 came.
Yeah. And where were you in the company's journey when you realized how big a problem COVID was going to be to the world?
Very early. So I was at NIH when SARS-CoV-1 came in 2000. And because of all this, NIH made huge efforts into looking into pandemic preparation. And the conception was that any day this H1N1 can convert into some beast and can come. So what kind of priority? So we were having a surveillance group who was analyzing it. And somehow, because different government policies, the most of the surveillance group was broken. Yeah. But still some of the surveillance group was available, that system was available, and which got first time in New York that so many deaths happened overnight without knowing the reason. And I was in Washington, D.C., and when I checked with my colleagues at NI, this was considered, and immediately I wrote back to my Secretary Department of Biotechnology in India, Government of India, that looks like this is a big problem. If 279 people have died in one night, it is a huge problem. And by that time, this SARS-CoV-2 genome was also published, and we were aware of, because my next door neighbor, Barney Graham, basically, who made the Moderna vaccine, he was working on mRNA, and because as I come from the field of vaccinology, I believe mRNA technology has a great future. It may not work for everything, but it has a great future because you can have to send a message only to make the protein. So body is acting as a fermenter or making the protein for you. And we interacted with NIH, and NIH says, yes, this is a good idea to go with. And we moved at that point.
You've developed a platform that you've now got emergency use authorization for, and it's being used now as a vaccine across India, and I'm guessing you've got plans for the future. How did you get to that platform so quickly? Were you already exploring that area, or did you do that in those first few months?
So we are already exploring with some of the partners, because I was looking this mRNA platform for cancer vaccines.
Right.
Or something like flu vaccine, because every flu season, when the sequence is known, then you have to work and all that. But when I saw the field, there were certain fundamental problems were there in the field. So there was a Grand Challenge program where Mr. Gates said that scalability is a problem. And the second, he said that ultra low temperature is a problem.
Right, yeah.
So yes, we understood the platform, we moved into the platform. And again, as every mRNA company has some litigation, we also have a litigation.
Yes.
And very openly, I want to say it. But we said, if we don't solve these two problems, scalability and low temperature, then this innovation will not be of value in low and middle income countries.
Yeah.
So we went into it, understand why the stability of DNA and mRNA is different, and we figured out there's one stupid OH group is there which is creating a problem, and because this, in presence of water, it breaks the phosphodiester bond, and if you remove the water from the preparation, then vaccine is stable. So we created a lyophilized vaccine, and which is today stable at 2 to 8, or you can say 25 degrees centigrade is stable. And this is the way the lyophilized vaccine has gone. So this is one part of the innovation. And the second part, we are waiting for the approval of the booster also. And here again, we have used a different technology to deliver the vaccine. This is intradermal delivery.
Yes.
Because there is a possibility that if nucleic acid vaccine goes into the vein, it creates some myocarditis. and that we experimentally demonstrated, other groups have experimentally demonstrated. So this technology, which we partnered with a group based in Boulder in US, they have PharmaJet. So there was a lot of demand because basically the dermis is the place where all the immune defense system is available.
Yeah.
But delivery was not there. So now we have completed the 3,200 volunteer trial for the booster. And hopefully this vaccine will be available in a couple of weeks
Oh,
for
fantastic.
Omicron variant.
Yeah, no, that is great. And you've managed to keep the dose quite small, haven't you?
So this is self-amplifying mRNA. So Moderna and Pfizer were on the non-replicating, but we decided to go on self-amplifying. This was against the norms because once mRNA vaccine is given, very quickly it fade away. So antigen is not available for a longer time, but in our case, looks like antigen is available because self-amplifying amplify itself with time and then it wanes off. So this is also adding some value on the memory cell or T cell immunology, not only the humoral, and that looks like we are doing good.
No, I mean, what a great story. And I'm guessing that this platform is not just a COVID-19 platform. You're thinking that there are other things that you can do with this.
Yes, certainly. And as I said, that we innovate for our local needs, and then this innovation can go outside. So at present, one of the big target is like dengue. Can we develop this platform for a single dose HPV vaccine?
Right. Mm-hmm.
So compliance could be much easier and much effective. So hopefully this is going to help in many diseases to develop the preventive measures at affordable cost.
Yeah, 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 and that's a really big piece, isn't it? I love the phrase you've been quoted as saying about democratizing the use of these products, and I think that's a really positive development that you've just described there. And as you look to the future, Dr. Singh, what's your biggest hope for the things that you've done with Gennova and the sort of the science that you've created?
So I feel the most important thing is what kind of mission an organization drives for a longer term of success. The first thing, fundamentals would be absolutely right, that for what organization is there. And we say that we are there to transform the healthcare.
Yeah.
So can we what are the basic fundamental problems? So vaccinology is one of the basic fundamental problems because there are many diseases, possibility to get readiness for pandemic. And then you should have manufacturing capabilities and you should collaborate globally. Because I believe that we do not know that what a human race will achieve in next 100 years. But when we see back, we have achieved in a century great things.
Yeah.
Just like we were having a 64 kilobyte floppy disk.
Yes.
You have to
Yes.
load and you have to DOS and everything you have to do. And now you can have a couple of GB on a stick.
Mm-hmm.
So this is just a question of imagination. Human being is only limited by imagination, nothing more. And where our imagination should go, our imagination should go to solving big problems in healthcare. Just like tuberculosis is a very big problem. Can we solve it?
Yes. Yeah.
Can we eradicate cervical cancer by HPV vaccine, which should be democratized because at present the 9-valent vaccine is available in the West, but in most of the countries in the world, divalent or tetravalent is available because cost is not affordable.
Yeah.
So can we bring the best innovation of 9-valent into affordable range? And then can we add something more on it? So compliance of taking the vaccine, adherence to vaccine, its effects would be. So those are the kind of imagination, if you ask me, I have. Can we solve some big problems?
Yeah.
Can we eradicate the cervical cancer from these countries, which is one of the major cause of the death? Last year, 390 million cases was dengue. Can we make a vaccine which prevent this? So these are the local problems we are trying to work around.
I love that vision. It's a great vision, and I'm very supportive of you in what you're saying there. That's great. Just one last question before we finish. When you're doing, I mean, the things that you've described that you've done, I mean, it sounds a very committed life that you have. Do you ever stop? What do you do when you stop? What are the kinds of things you do to relax from this daily dreaming of the future that you've just described?
I work and work, and work relaxes me.
Well, Dr. Singh, I think the world will be a better place for you doing that. So thank you for doing that. And can I say what a pleasure it's been talking to you on LabYak. It's just great hearing that story, and I've really enjoyed hearing how you got from where you were to where you are now. And your dream of the future is one I think that all of us can share. So thank you for that. And thanks again for being on LabYak.
Pleasure is mine. Thank you very much.